Hello everyone. Welcome to week 2 of Lenore Ryan's 10-week COVID class. My name is Daniel Grimm. I'm an assistant professor of biology in the School of Natural Sciences here at LR. And I want to thank Drs. Taylor Newton and Devin Fisher for inviting me to participate in this class. During the regular semester, I teach a course on the fundamentals of microbiology. In that course, we explore the nature of a variety of microbes, including bacteria, fungi, protozoa, algae, and viruses. Given that the current pandemic is caused by a virus, I thought it might be helpful to review exactly what viruses are and explore the conditions under which they thrive. We hear a lot of talk in the media and on the internet about the COVID-19 virus, the formal name of which is SARS-CoV-2. But I imagine there are those of you in the audience who might want to know a little bit more about what viruses actually are, what they are made of, how big they are, where they can be found in the environment, if they're even alive, what kind of cells they might infect, or how they replicate and spread. So my goal in this lecture is to introduce the fundamental characteristics of the group of microbes we call viruses, so that we might better understand the specific nature of the pathogen responsible for the disease we know as COVID-19. The more we know about the physical and chemical characteristics of a pathogen and how it behaves in a host, the better we can plan therapeutic interventions to stop the spread of an infectious disease. The objectives of this lecture are first to describe the general characteristics of viruses, then to highlight the differences between viruses and other microbes. We will then talk about the chemical and physical structure of both enveloped and non-enveloped viruses, giving special attention to the structure of the novel coronavirus responsible for the current pandemic. Finally, we'll take a look at some micrographs that illustrate the damage viruses can do to our cells during an infection. Note that at the bottom of this slide, I've included a reference to the text that we use in our microbiology course at LR. It is entitled Microbiology and Introduction. This is a very informative and easy-to-read book in its 13th edition. I recommend it to anyone interested in reading further about viruses or microorganisms in general. I want to start by defining just a couple of terms that we encounter every day and that we will use throughout this lecture. I think most of us are familiar with these terms, but I've found through the years that students sometimes confuse the concept of disease with the causative agent of that disease. So let's define both of these in light of the coronavirus pandemic we are experiencing. COVID-19, as most of you are aware, is a term applied to the actual disease caused by the novel coronavirus. This acronym comes from the first syllables of corona and virus. The D represents disease, and the 19 is from 2019, the year in which this disease was first described. SARS-CoV-2 refers to the causative agent of the disease, a novel virus belonging to a family of viruses called coronaviridae. The SARS portion of the virus name refers to a medical disorder associated with the disease, severe acute respiratory syndrome, and the number 2 indicates that this is the second such coronavirus we've seen that causes the syndrome. The first was a SARS coronavirus responsible for a deadly outbreak of this syndrome in 2003. Okay, on to the basics. A good definition of viruses, I think, is the descriptive one you see here. Viruses are submicroscopic, parasitic, filterable agents consisting of nucleic acid surrounded by a protein coat. What I mean by these descriptors is, first, that viruses are too small to be seen with a conventional light microscope. These instruments max out at magnifications of about 1,000 to 1,200. Viruses are so small that they require a more powerful electron microscope in order to see them. Second, viruses are parasitic. In fact, they are obligate intracellular parasites. They are completely inert outside their host cell, upon which they are totally dependent for all their metabolic and reproductive functions. With very few exceptions, viruses are much smaller than bacteria and will pass through the pores of special filters designed to retain bacteria, so they are filterable agents. Finally, their structure is very simple. They are composed of nucleic acid, either DNA or RNA, surrounded by a protective coat of protein. In this respect, they can be viewed as highly complex chemical entities, and not the living organisms as we normally define them. Viruses were first discovered when researchers working with tobacco mosaic disease, or TMD, would grind up diseased plant materials in water and pass the material through a bacteriological filter. They suspected that the causative agent of TMD was a bacterium, but they found that the bacteria-free liquid filtrate they generated would cause the disease when injected into another tobacco plant. In fact, the term virus is Latin for liquid poison. As researchers continued their work on this unknown ultra-small pathogen, the development of electron microscopes in the 30s and 40s allowed them to actually see viruses and begin to describe their characteristics. Viruses are not composed of cells. They are totally dependent on a host cell and are completely inert outside that cell. They are inanimate particles. Since viruses are not cellular, they don't have cell walls or plasma membranes. And unlike cell-based organisms, they don't possess both DNA and RNA as their genetic material. They have one or the other. Viruses do not have cellular structures like mitochondria that would enable them to produce energy and store it as adenosine triphosphate, or ATP. Nor do they have ribosomes required for the synthesis of structural proteins and enzymes. The host cell for the virus must provide the machinery for all these functions. During the early days of antibiotic discovery and development, we learned that viruses are not sensitive to antibiotics, and antibiotics have no effect on the spread of viruses during an infection. We also discovered that, unlike bacteria, viruses do not reproduce by binary fission, a process by which a bacterium will replicate its DNA, elongate, and divide into two daughter cells. Instead, they go through a process of binding to and then invading a host cell, separating their nucleic acid from its protein coat, a process called uncoating, and then relying on the host cell to replicate viral genes, synthesize proteins, and assemble the virus particles prior to expelling the newly formed viruses from the host cell. So, viruses are entirely dependent on the metabolic processes of their host cell for their continued existence. And their host cell range is quite extensive. Different viruses are capable of infecting fungi, the yeasts and molds, bacteria, vertebrate and invertebrate animals, algae, protozoa, and higher plants. Virtually any cell is a potential host for a virus. Most of what we know about virus replication we've learned by studying a group of viruses that infect bacteria exclusively. These are the bacteriophages, a term that literally translated means bacteria eaters. Research in this area has also led to the potential use for these bacteriophages to treat infections caused by antibiotic-resistant bacteria. The size range for viruses is typically between 20 and 1,000 nanometers. A nanometer is a millionth of a millimeter, or about 25 millionths of an inch. Bacteria are larger and usually fall between 1,000 and 8,000 nanometers in diameter. So, electron microscopy is required to see the vast majority of viruses. I really like this slide because it gives us an idea of the size of viruses compared to bacteria and our own cells. On the left is a light micrograph of a human cheek cell with the surrounding plasma membrane labeled PM and the nucleus labeled N. The small blue rod-shaped cells indicated by the arrow are bacteria commonly found in our mouths that are attached to the surface of the cheek cell. If we take the single bacterium on the lower right side of the cell and artificially enlarge it to the size shown on the electron micrograph on the right, we could begin to appreciate the very small size of viruses. In the picture on the right, you will notice the many viruses, termed coliphages, covering this rod-shaped bacterial cell we know is E. coli. A single coliphage virus is circled in red and highlights the size difference between bacteria and viruses. This is an illustration from the textbook I use in microbiology that shows the size range of viruses, from the very small poliovirus at 30 nanometers to the large Ebola virus coming in at over 900 nanometers. I've added the SARS-CoV-2 coronavirus in the lower right-hand corner for comparison. It typically has a size range between 50 and 200 nanometers. By the way, the first person to actually see and describe coronaviruses was a Scottish researcher who specialized in electron microscopy named June Almeida. A photo of her and one of her early electron micrographs of a coronavirus that infects chickens is shown on this slide. You can certainly see similarities between Almeida's micrograph and the image of the novel coronavirus responsible for the current pandemic. When we talk about virus structure in microbiology class, we use the term virion to refer to the complete, fully developed infectious virus particle. During the replication process, viruses disassemble in their host cell and exist as individual parts of the whole virus. So the term virion is used only for the complete virus that can be found outside the host cell. During the infection process, it is the vehicle of transmission from one host cell to another, or person to person, and from a surface to a host cell. Virologists and physicians sometimes refer to groups of viruses that have similar characteristics by specific names based on the virus classification. For example, viruses can be organized based on which nucleic acid they possess or what kind of protein coat they have surrounding that nucleic acid. Viruses can have DNA or RNA as their genetic material, and depending on the species of virus, the nucleic acid can exist as single or double-stranded, linear or circular. A virus is often referred to as a DNA virus or an RNA virus. Some examples of DNA viruses are shown on this slide. They include the adenoviruses, one of the causes of the common cold, papillomaviruses, which are implicated in genital warts and cervical cancer, and the causative agents of smallpox, chickenpox, and hepatitis B. Viruses which contain RNA as their genetic material include the influenza virus, hepatitis C virus, rabies and Ebola viruses, and the coronaviruses, including SARS-CoV-2. So, by this classification scheme, the virus responsible for the current pandemic, the SARS-CoV-2 virus, is a linear, single-stranded RNA virus with one of the largest genomes of all the viruses, indicating it contains an abundance of genes regulating its attachment and entry into host cells and its subsequent replication and survival. But viruses can also be grouped according to what surrounds or covers their genetic material. Recall that viruses are nucleic acids surrounded by a protein coat. This protein coat is termed capsid and is often composed of smaller units we call capsomeres. This is an electron micrograph and crystalline model of the adenovirus, one of the viruses responsible for the common cold. If you look closely at the capsid, you'll see it is composed of multiple smaller subunits. These are the capsomeres. I've drawn a red circle around a single capsomere to highlight its shape and size. In some viruses, the capsid is surrounded by an additional layer of material composed of fatty acids combined with proteins and sometimes carbohydrates. This additional covering is called an envelope. An example of a common envelope virus is the influenza virus. In this transmission electron micrograph, you can see the helical capsid, seen here folded in an upside-down U shape, surrounded by the viral envelope. The envelope of the influenza virus and some other envelope viruses contains protein projections called spikes. These spikes have a variety of functions depending on the species of virus, including attachment to the host cell and inducing clumping of red blood cells during an active infection. In this micrograph, you can see more clearly the protein spikes of the envelope surrounding the influenza virus. The SARS-CoV-2 virus is also an envelope virus. Like the influenza virus, the COVID virus envelope is covered with protein spikes which allow the virus to attach to our lung epithelial cells. Attachment is the first step in viral entry into the cells during the infection process. In fact, we now know that the envelope of the COVID virus contains several protein projections, each with a different function. The spike protein, or S protein, enables the virus to attach to the host cell. It binds to a receptor on the surface of our lung cells called the ACE2 receptor. The small envelope protein, or E protein, plays a role in the assembly of newly formed viral particles and the release of the virus from the host cell during the replication process. The nucleocapsid, or N protein, surrounds the genetic material of the virus, in this case RNA, and protects it from destruction by the host cell defense mechanisms. Finally, the M, or membrane protein, is the most abundant protein in the envelope and gives the viral envelope its shape and structural rigidity. We know that only some viruses have an envelope. Viruses that lack this feature are called non-envelope viruses. One of the most common virus structures is the polyhedral capsid without an envelope. The micrograph on this slide shows the non-enveloped rotavirus, the most common cause of diarrhea in infants and children worldwide. So we can talk about DNA and RNA viruses and enveloped and non-enveloped viruses, but we can also group viruses based on the morphology or structure of their protein coats, their capsids. For example, helical viruses are long, rod-shaped particles that can be rigid or flexible. The Ebola virus is a type of helical virus that has a flexible filamentous capsid. Notice the distinctive Mickey Mouse ears at one end of the helical rod. As I mentioned earlier, a very common capsid structure is the polyhedron, an icosahedron consisting of 20 triangle-shaped faces. This is the capsid structure found in adenoviruses and the poliovirus. Viruses with envelopes exhibit a type of morphology that is roughly spherical. This is due to the nature of the envelope. Both helical and polyhedral viruses can have an envelope. The micrograph here shows the polyhedral herpes simplex virus surrounded by an envelope. By the way, you can really see the individual capsomeres in this picture. Lastly, we have the viral capsids with complex structures. These are some of the most remarkable viral morphologies in nature. This is a scanning electron micrograph of a virus that infects bacteria exclusively. It is known as the T4 bacteriophage. It has a polyhedral capsid with a helical tail sheath. The individual parts of the bacteriophage are labeled on this slide and include the tail fibers responsible for attachment to the bacterial cell wall that enables the virus to inject the DNA from its capsid through the tail sheath and into the bacterium. It is ultimately very bad news for the bacterium when these complex viruses infect it. Here, the bacteriophages are simultaneously trying to enter at one end of the bacterial cell, while other newly replicated viruses inside the bacterium will eventually destroy it by bursting out of the cell. At the beginning of each semester, I scan for news articles about microorganisms that I can share with my introductory microbiology classes. We'll discuss current events related to these particular microbes, why they're in the news, how do these news articles relate to our class. This last semester, I ran across a few news items about a novel pathogenic virus that originated in China and seemed to be spreading within that country. Our classes decided to follow these news briefs during the course of the semester. Little did we know what effect this one mutated virus would have on the very fabric of our daily routines, nor the toll it would take and is still taking on human lives. Looking back at these late January news stories that I shared with my classes, the CDC notice confirming the existence of U.S. cases takes on an ominous tone for me. But at the time, we were still not getting reports of widespread infection or death in China. Of course, all that would soon change. So, what is the novel coronavirus? And based on the general characteristics and descriptions of viruses that we've reviewed so far, exactly where does this new virus fit in all this? The SARS-CoV-2 virus is an envelope virus with large protein spikes used for attachment to the ACE2 receptor on the surface of cells in our nasal passages, lungs, and intestines. It has a size range of 50 to 200 nanometers in diameter, and about 150 million average-sized coronaviruses would fit on the head of a straight pin. The novel coronavirus is an RNA virus containing a very large, linear, single-stranded RNA molecule. It has a helical capsid residing within a lipid protein envelope, which gives it a roughly spherical shape. It takes its name from the word corona, which is Latin for crown or halo. The similarity between the virus's appearance under the electron microscope and the corona around the sun that is visible during a total eclipse is the origin of the name coronavirus. The family Coronaviridae is an RNA virus family that contains 7 distinct virus strains, including 4 that are responsible for the common cold, plus the SARS, MERS, and SARS-CoV-2 viruses. Coronaviruses were first isolated in the 1960s from nasal swabs taken from people suffering from the common cold. In 2003, a unique coronavirus caused an outbreak of a pneumonia-like disease given the name Severe Acute Respiratory Syndrome, or SARS. The virus responsible for this syndrome originated in China and was believed to be a spillover strain that mutated from a virus in civet cats. Diseases that are spread from animals to humans are called zoonotic diseases. More than 8,000 people from 29 different countries contracted this particular zoonotic disease. The high mortality rate resulting from this infection was alarming at the time. A few years later, in 2012, a coronavirus was implicated in another respiratory disease with an even higher mortality rate. The Middle East Respiratory Syndrome virus was thought to have originated in bats, then jumped to camels, and from camels to humans. According to the World Health Organization, about 2,500 cases have been reported since 2012, and approximately 35% of those who are diagnosed with MERS will succumb to the infection. Then, in late 2019, an outbreak of a new viral pneumonia was reported in Wuhan, China. The origin of the outbreak was believed to be an outdoor seafood and animal market in the city of Wuhan. The viral agent responsible for the outbreak was soon identified as a coronavirus that was a spillover strain originating in bats. As we soon learned, there is of course human-to-human transmission of the disease that became known as COVID-19. Although direct transmission of the virus may occur when coming into contact with saliva or secretions of an infected person, the principal means of transmission is by airborne droplets containing the virus coming into contact with mucous membranes in the eyes, nose, or mouth of a susceptible person. This gives the virus access to the respiratory system of the new host. Signs and symptoms of COVID-19 include a dry cough, fever, and difficulty breathing. The infection is so new, though, that we have very few resources for treating it. Since it's a viral infection, antibiotics don't work. There is evidence, though, that the drug remdesivir can shorten the duration of the illness by a few days in some patients. In the meantime, several vaccines to prevent infections are under development. We'll talk a little more about vaccine development when we meet again on Thursday. The more we know about a pathogenic virus like SARS-CoV-2, the better equipped we are to devise a treatment for the disease caused by that virus. But to study a virus, we have to be able to grow it in the laboratory. So how do we do that? In order to isolate specific viruses from a sample and grow them for the purposes of research or vaccine production, viruses have to be provided with living cells. Since they are completely inert outside their host cell, we can't grow viruses in the kinds of chemical media we use to grow cell-based microorganisms, such as bacteria or fungi. What we can do is grow bacteria on artificial media and then use bacteria cells to propagate viruses. In fact, extensive research on how viruses replicate and how they can incorporate some of their genes into the genetic material of the host cell has been done by growing bacteriophages in bacterial cells. But if we want to study animal viruses like the COVID virus, we can't use bacteria. Instead, we grow them in living animals, embryonated eggs, or cultures of animal cells grown in the laboratory under controlled conditions. Growing viruses in living animals can be difficult and expensive, but embryonated eggs offer an inexpensive and convenient source of cells in which to grow them. Multiple membrane surfaces within the egg are capable of sustaining viral growth. In fact, prior to the administration of some vaccines, your physician may ask you if you are allergic to eggs. That's because the most efficient way to produce some viruses for vaccine production is to grow them in embryonated eggs. Many viruses used in vaccine manufacture are grown in cell culture. Specific cell types are matched to specific viruses to optimize virus replication. These cell lines can be grown in the laboratory much like we grow bacteria. The cells grow in a single layer at the bottom of a plastic or glass culture flask. Not only can we grow viruses for harvest, but we can study the effect a virus infection has on cells by observing the microscopic changes in cell appearance during viral growth in a culture. A viral infection in cell culture often leads to the deterioration and eventual destruction of the host cells. The SARS-CoV-2 virus has been shown to grow quite well in a few cell lines, including human airway epithelial cells, 2 cell lines derived from kidney tissue, hamster ovary cells, and human bone marrow cells. The image on the right of the slide is a light microscope view of Vero E6 cells growing in a culture flask. You can see a single kidney cell surrounded by the red oval. By the way, I've included 2 links at the bottom of the slide to videos showing COVID growth in cell cultures. During their growth in cells, viruses can cause changes in those cells that we can see under the microscope. These visible changes are called cytopathic effects. A viral infection can result in the interruption of cell division, the release of lysosomal enzymes that damage the interior of the host cell, or the accumulation of what we call inclusion bodies in the cytoplasm or nucleus of the cell. These inclusion bodies are sometimes made of pieces of virus left over from viral replication, and they vary in size, shape, and location depending on the species of virus causing the infection. For example, a cytomegalovirus infection is characterized by the production of inclusions in the nucleus of the host cell that resemble owl's eyes. A diagnosis of cytomegalovirus infection can be made based on this particular inclusion morphology. Viruses can cause host cells to convert from their typical flat configuration in cell culture to an abnormal spherical shape in a process we call rounding up. This cytopathic effect is typical of some viruses, including the COVID virus. COVID virus infection can also cause neighboring cells in the culture to fuse with each other. The nuclei from these individual cells will then accumulate in a giant fused cell called a syncytium. This micrograph illustrates the rounding up effect that some viruses have on a cell culture. The label indicates a rounded cell caused by the Rous sarcoma virus. This micrograph shows the formation of a syncytium, the giant multinucleated cell caused by introducing the COVID virus to a kidney cell culture. A normal uninfected kidney cell is circled in red. Viruses can also cause the outright death of a cell, or even genetically transform a cell, resulting in unregulated cell growth characteristic of some cancers. In cell cultures, the SARS-CoV-2 virus has been shown to promote cell rounding, detachment of cells from the flask surface, syncytia formation, cell degeneration, and cell death. The take-home message here is that virus infections in laboratory cell cultures can give us an idea of the possible mechanisms by which a virus might damage the cells in our bodies during an infection. In addition, these cytopathic effects are often characteristic of specific viruses, and so they are a useful diagnostic tool for clinicians. I'll wrap up our lecture with this final slide, showing an electron micrograph image of the COVID-19 virus emerging from the surface of an infected cell. I hope this general review of viruses was beneficial to you and provided some insight into the nature of the SARS-CoV-2 virus. At the beginning of our Thursday evening session, we'll have a brief review of the various approaches to vaccine development and then move on to the Q&A session. I'll end here by stating something that you already know, and that is that knowledge is power. And with respect to the current pandemic, the more we learn about the structure of the COVID-19 virus and its behavior in host cells, the greater our chances of developing successful therapies for the treatment or prevention of this disease. I'm greatly encouraged by the impressive worldwide effort in drug and vaccine development that has taken place over the last 5 months, and I'm optimistic that these extraordinary efforts will soon result in the suppression or elimination of this disease. Thank you for listening, and I'll see you on Thursday.